Live from WLMK, a Keenan broadcast network, it's Microbiology in the News, with your hosts,
Stacy Montalongo,
Brendan Sharrow,
and Ben Jenkins, who will be here in just a few minutes.
And I'm Crystal Baumgartner.
Good morning. Scientists reveal surprising picture of how powerful antibody neutralizes HIV. Researchers at the Scripps Research Institute have uncovered the surprising details of how powerful anti-HIV antibody grabs hold of the virus. The findings that were published in Science Express on October the 13th, 2011 highlight a major vulnerability of the HIV virus and suggest a new target for the vaccine development. Researchers from the current team recently isolated the new antibody and 16 others from the blood of HIV-infected volunteers in work they reported online in a journal, Nature, on August 17, 2011. Since the 1990s, Burton, who works with Dennis Burton, a professor at the Scripps Research Institute, and other researchers have been searching for such broadly neutralizing antibodies against HIV. Antibodies work against the many various strains of the fast mutating virus, and by now they have found more than a dozen. The PGT-128, the antibody described in the new report, can neutralize about 70% of globally circulating HIV strains by blocking their ability to infect cells. The structure of the PGT-128 joined to its binding site on molecular mock-ups of the virus. With these structural data, and by experimentally mutating and altering the viral target site, they could see that the PGT-128 works in part by binding to glycans on the viral surface. Thickets of these sugars normally surround the HIV's envelope protein, shielding it from the attack by the immune system. Nevertheless, PGT-128 manages to bind two closely spaced glycans, and at the same time reaches through the rest of the glycan shield to take hold of a small part of the structure. The antibody binds to the GP120 in a way that presumably disrupts the ability to lock onto human cells and infect them. Researchers hope to use the knowledge of these antibodies binding sites on HIV to develop vaccines that stimulate a long-term, perhaps lifetime, protective antibody response against those same vulnerable sites. We probably need multiple targets on the virus for a successful vaccine, but certainly the PGT-128 shows us a very good target, said Professor Burton.
Good story. That was a good story.
Thank you. I enjoyed doing that one.
A research team at the National Institute of Standards and Technology has provided its first look at the genetic structure that may play a critical role in the reproduction of the infectious salmon anemia virus, more commonly known as the fish flu. While there is a vaccine for the virus, it must be administered by injection, a task that is both cumbersome and economically impractical. A drug or vaccine prevents...
Sorry, big wreck, big fiery mess. It was bad. Sorry.
And now we have Anchor Ben Jenkins.
Hey. Go ahead. Sorry.
Alright. A drug or vaccine that prevents the spread of the disease by interfering with the virus's ability to replicate its genetic code would be far more practical for fish farmers and marine biologists to deliver. Robert Brinson, a scientist working at the Hollings Marine Laboratory in Charleston, South Carolina, along with Andrea Skazel and John Marino, working at the Institute for Bioscience and Biotechnology Research in Rockville, Maryland, knew from the scientific literature that the families of viruses that include both the many types of influenza, causes of yearly human flu outbreaks, and the infectious salmon anemia form a panhandle structure in their genomic RNA. In human influenza, the panhandles are known to interact with proteins that begin the process of copying and replicating the virus. Hypothesizing that the fish flu virus might function the same way, Brinson used high-resolution nuclear magnetic resonance spectroscopy and thermal melting methods to look at the genetic structure in the same region of the RNA. They found that the genome does appear to have a panhandle motif. The work provides the first experimental evidence and characterization of this panhandle motif that may function similarly in the fish virus as the green light for viral RNA replication processes. Brinson says the next step is to investigate the relevant proteins and how they interface with the RNA, what molecular features drive the protein and to recognize the RNA, how it is binding, and how it is interacting with the RNA. Brinson says this work may facilitate the development of new approaches for interfering with the replication of these viruses to mitigate the effects on the aquacultural salmon. It also demonstrates that the salmon virus can be used as an experimental model for understanding the replication machinery of human and other related influenza viruses.
So do we say bless you whenever they get the flu?
We do.
We do.
That's incredible.
Actually, I thought we said go to hell.
You're right, man. I forgot.
That's a good one.
It all sounds very techy.
Yes, it does.
Shoot.
Shoot, bless you. Oh, excuse me. Okay. All right, so in other news, Kawasaki disease linked to wind currents. First evidence that long-range wind transport of an infectious agent might result in human disease. Kawasaki disease is a severe childhood disease that many parents, even some doctors, mistake for an inconsequential viral infection. In fact, if not diagnosed or treated in time, it can lead to irreversible heart damage. After 50 years of research, including genetic studies, scientists have been unable to pinpoint the cause of the disease. Now, surprisingly, findings of an international team of scientists organized by Jane Burns, MD, Professor and Chief Division of Allergy, Immunology, and Rheumatology of the University of California, San Diego School of Medicine's Department of Pediatrics, and Rady Children's Hospital, San Diego, suggest that KD disease are linked to large-scale wind currents that track from Asia to Japan, also transfers from North Pacific. Our findings suggest that environmental trigger for Kawasaki disease that could be windborne. Burns says the paper will appear in the Nature Scientific Reports on November 10th. Signs of KD include prolonged fever associated with rash, red eyes, mouth, lips, tongue, and swollen hands and feet with peeling skin. I may have this. The disease causes damage to the coronary arteries in a quarter of untreated
I believe
children.
gonorrhea does that too, Crystal.
I should check that. It may lead to serious heart problems in early adulthood. There is no diagnostic test for Kawasaki disease, and current treatment fails to prevent coronary artery damage in at least 1 in 10 to 20 children and death in 1 in 1,000 children. While seasonality of the disease has been noted in many regions, particularly in Japan, the country of the highest incidence for KD, the search for factors that might contribute to epidemics and fluctuations in KD occurrence has been exclusive. A study of KD cases in Japan since 1970 showed 3 dramatic nationwide epidemics, each lasting several months and peaking in April 1979, 6,700 cases, May 1982, 16,100 cases, and March 1986, 14,700 cases. These 3 peaks represent the largest KD epidemic events ever recorded in the world. To investigate a possible influence from large-scale environmental factors, researchers include Daniel Keenan, Climate Atmospheric Science and Physical Oceanography, KASPO, at Scripps Institution of Oceanography in La Jolla, and Xavier Rodo and Joan Ballister, the Institute of Catala di I'm pretty sure I went there one year. And in Barcelona, Spain, investigated a set of atmospheric and oceanographic graphic measures which revealed a link to pressure patterns and associated wind flow from the surface to mid-atmospheric levels during the summer months prior to onset of the epidemics. The Japanese data set revealed that a low number of KD cases were reported prior to the epidemics, a period coinciding with the winds from the south which blew across Japan from the Pacific Ocean during the summer months, said Rado, the study's first author. However, the numbers rapidly mounted all over Japan when winds turned and blew from the northwest, a trajectory from the Asian continent. After the peaks, the winds again shifted, blowing from the south when the number of cases again decreased. Importantly, subsequent to the 3 epidemics, years with increased numbers of Kawasaki disease cases in Japan were significantly associated with enhanced local northwesterly winds as a result of low pressure centered to the north, said Keenan. To access weather, such variations in wind patterns were associated with KD cases fluctuations on the other side of the North Pacific. Similar analysis were conducted for San Diego. According to the scientists, the atmospheric connection from continental Asia to Japan and San Diego is intermittent and can take different routes. However, it was possible from the analysis to identify the major anomalous yearly peaks of KD cases occurring in San Diego from 1994 to 2008 as belonging to 2 main atmospheric configurations. In fact, the major fluctuations in KD case numbers in Japan, Hawaii, San Diego were linked to a seasonal shift in the winds that exposed Japan to air masses from Central Asia. One key pattern simultaneously exposed Hawaii and California to air masses from the western North Pacific. The link to wind currents, which can cross the Pacific in less than 1 week, may explain why KD case numbers reported in Japan, San Diego, and Hawaii show a nearly synchronized seasonal peak in disease activity from November to March. Additional contribution.
Hey, little girl. We're on the timeline here.
Additional contributors study include Martin John School of Medicine in Honolulu, Hawaii.
Okay, now, Ben.
Sorry to hear about the disease you may or may not have. Spoiled
You
food.
need to edit.
Spoiled food, soon a thing of the past. The Norwegian researchers have developed new methods to identify potential sources of contamination. Microorganisms such as bacteria, yeast, and molds can cause food spoilage and deterioration during food production. They could be lurking in the tubes that convey milk and to their cartons, lying in wait on the floor, or even floating in the air where sausages are being packaged. And they pose to attack or contaminate our food at any time. Troubleshooting during production in the food industry's current approach using observation and testing to locate the causes of food spoilage. Using a spectrometer, the researchers are able to detect undesirable microorganisms in finished products and trace them back to the various steps in the production process. Scientists have long used Fourier transform infrared (FTIR) spectroscopy as a method of chemical analysis, and for the last 2 decades for identifying bacteria as well. But no one had so far used this method for identifying food contamination. Researchers at NOFOM and MAT have taken spectral readings of various microbes collected from a wide variety of foods, including juice and milk. Each microbe has its own unique spectral profile, which functions much like a fingerprint that was used for identification purposes. They stored 20,000 spectral profiles of different microbes, and wherever they found an unknown microbe, they took a spectral reading of it and compared it to a profile of the same. The group says there are a number of methods used for locating sources of contamination in the food industry, but they are convinced that there is the most cost-effective solution. For more info, go to the Research Council of Norway's website.
So I guess that means Miss Keenan's going to finally get to eat all our celery.
Yep, I think so.
Well, there you have it. And now we're going to talk about computing blocks. You talked about the fact that we had something high-tech a while ago. Computing blocks are building blocks created from bacteria and DNA. On October 18, 2011, scientists successfully demonstrated that they can build some of the basic components for digital devices out of bacteria and DNA, which could pave the way for a new generation of biological computing devices. The research from the Imperial College of London have demonstrated that they can build logic gates, which are used for processing information in devices such as computers, microprocessors, out of harmless gut bacteria and DNA. These are the most advanced biological logic gates ever created by scientists. They state that the logic gates are the fundamental building blocks in the silicon circuitry that our entire digital age is based on. Without them, we could not process digital information. Now that we have demonstrated that we can replicate these parts using bacteria and DNA, we hope that our work could lead to a new generation of biological processors whose applications in information processing could be as important as their electronic equivalents. Although still a long way off, the team suggests that these biological logic gates could one day form the building blocks in microscopic biological computers. Devices that may include sensors that swim inside the arteries, detecting a buildup of harmful plaque and rapidly delivering medication to the affected zone. Other applications may include sensors that detect and destroy cancer cells inside the body and pollution monitors that can be deployed in the environment, detecting and neutralizing dangerous toxins such as arsenic. The scientists have constructed a type of logic gate called an AND gate, from the bacteria Escherichia coli, which is normally found in the lower intestine. The team altered the Escherichia coli with modified DNA, which reprogrammed it to perform the same switching on and off processes as the electronic equivalent when stimulated by the chemicals. The next stage of the research will see the team trying to develop a more complex circuitry that comprises of multiple logic gates. One of the challenges faced by the team is finding a way to link multiple biological gates together, similar in a way in which electronic logic gates are linked together to enable a complex processing to be carried out. I guess that means if your computer goes bad, you can just poop on it.
That should be your catchline.
Wasn't expecting that one.
Oh, you're in trouble.
Now, the scientists at the Salk Institute for Biological Studies and their collaborators found that tweaking a gene known as PGC-1, which is also found in human DNA, in the intestinal stem cells of fruit flies, delayed the aging of their intestine and extended their lifespan by as much as 50%.
Wow.
Fruit flies and humans have a lot more in common than most people think. There is a tremendous amount of similarity between a human small intestine and a fruit fly's intestine. The findings of the study, which was a collaboration between researchers at the Salk Institute for Biological Studies and the University of California, Los Angeles, were published online in Cell Metabolism. That's what I said. That's what I said. No, I didn't. I said metabolism. Metabolism. All right. While little is known about the biological mechanisms underlying this phenomenon, studies have shown that the cells of calorie-restricted animals have greater numbers of energy-generating structures known as mitochondria. In mammals and in flies, the PCG-1 gene regulates the number of these cellular power plants, which converts sugars and fats from food into the energy for cellular functions. The flies have a short lifespan, allowing the scientists to study aging and longevity in ways that aren't as feasible in longer-lived organisms such as mice or human. The researchers found that boosting the activity of the PGC-1, the fruit fly resulted in greater numbers of mitochondria and more energy production in the flies. The same phenomenon seen in organisms and on calorie-restricted diets.
Interesting. Now that I see it, you do kind of remind me of a fruit fly.
Good.
Oh yes. Honey can reverse antibiotic resistance. Manuka honey can be an efficient way to clear chronically infected wounds and could even help reverse bacterial resistance to antibiotics, according to research presented at the Society for General Microbiology Spring Conference in Harrogate. Professor Rose...
Stop that.
Manuka honey. Studies suggest honey can reverse antibiotic resistance. Manuka honey could be an efficient way to clear chronically infected wounds and could even help reverse bacterial resistance in antibodies. Looking at how Manuka honey interacts with 3 types of bacteria that commonly infest wounds, such as Group A, Streptococcus, and MRSA, found that honey can interfere with the growth of these bacteria in a variety of ways and suggest that honey is an attractive option for the treatment of drug-resistant wound infections. Honey has long been acknowledged for its antimicrobial properties. Traditional remedies containing honey were used in the topic treatment of wounds by diverse ancient civilizations. Our findings with Streptococcus and Pseudomonads suggest that Manuka honey can hamper the attachment of bacteria to tissues, which is an essential step in the initial of acute infections. Initiation of acute infections. Inhibiting attachment also blocks the formation of biofilms, which can protect bacteria from antibodies and allow them to cause persistent infections. Other work in the lab has also shown that honey can make MRSA more sensitive to antibiotics, such as oxacillin, effectively reversing antibiotic resistance. This indicates that the existing antibiotics may be more effective against drug-resistant infections if used in combination with Manuka honey.
It's very interesting.
Honey.
Darling.
Foodborne bacteria causes potentially fatal heart infection. Researchers at the University of Illinois at Chicago College of Medicine have found that particular strains of a foodborne bacteria are able to invade the heart, leading to serious and difficult to treat heart infections. The bacteria Listeria monocytogenes is commonly found in soft cheeses and chilled ready-to-eat products. For healthy individuals, Listeria infections are usually mild, but for susceptible individuals and the elderly, infection can result in serious illness usually associated with the central nervous system and the placenta and developing fetus. About 10% of serious Listeria infections involve a cardiac infection, according to Nancy Freitag, Associate Professor of Microbiology and Immunology and Principal Investigator on the study. These infections are difficult to treat, with more than 1/3 proving fatal, but have not been widely studied and are poorly understood. Freitag and her colleagues obtained a strain of Listeria and had been isolated from a patient with endocarditis, or infection of the heart. The researchers were interested in determining whether patient predisposition led to heart infection or whether something different about the strain caused it to target the heart. They found that when the infected mice with either the cardiac isolate or the lab strain, they found 10 times as much bacteria in the hearts of the mice infected with the cardiac strain. In the spleen and liver organs that are commonly targeted by Listeria, the levels of bacteria were equal in both groups of mice. Further, the researchers found that while the lab strain infected group often had no heart infection at all, 90% of the mice infected with the cardiac strain had heart infections. Listeria is actually pretty common in foods, says Tag, and because it can grow at refrigerated temperatures, as foods are being produced with longer and longer shelf life, Listeria infection may become more common. In combination with the aging population and more susceptible to serious infection, it is important that we learn all we can about these deadly infections. You can go online to the Journal of Medical Microbiology to find out more. Anybody want any cantaloupe?
What wasn't that? Isn't that what that was about? The cantaloupe? Wasn't that the listeria?
I brought some cantaloupe for you guys.
Okay, no,
It's been sitting out
good.
for a couple days, but
I'm
it's
good.
good.
I'm good. All right, fine. And now on the lighter, a little bit lighter, this next story that I have. How a zebra gets its stripe. A simple genetic circuit. On October 13, 2011, many living things have stripes, it was stated. But the developmental processes that create these and other patterns are complex and difficult to untangle. Now a team of scientists has designed a simple genetic circuit that creates a stripe pattern that they can control by tweaking just a single gene. With the genes taken from one species of bacterium and inserted into another, scientists from the University of Hong Kong assembled a genetic loop from two linked modules that senses how crowded a group of cells has become and responds by controlling their movement. One of the modules secretes a chemical signal called acyl homoserine lactone, or AHL. As the bacterial colony grows, AHL floods the accumulating cell, causing them to tumble in place rather than to swim. Stuck in the agar of their dish, they pile up. Because AHL doesn't diffuse very far, the few cells escape and swim away to begin the process again. Left to grow overnight, the cells create a target-like pattern of concentric rings, as we can see here, of the crowded, dispersed bacterial cells. By tweaking just one gene that limits how fast and far cells can swim, the researchers were able to control the number of rings the bacteria made. Although individual bacteria are single cells, as colonies, they act like multicellular organisms, sending and receiving signals to coordinate the growth and other functions of the colony. That means a fundamental rules that govern the development of these patterns could well apply to critical steps in the development of other organisms.
Cool. So is it white with black stripes or black with white stripes?
I don't know. They'll have to tell us that later.
One of the things that makes inhalation of anthrax so worrisome for biodefense experts is how quickly a relatively small number of inhaled anthrax spores can turn into a lethal infection. By the time an anthrax victim realizes he or she has something worse than the flu and seeks treatment, it's often too late. University of Texas Medical Branch at Galveston researchers have found new allies for the fight against anthrax, known as natural killer cells. They are a part of the immune system normally associated with eliminating tumor cells and cells infected by viruses. People become ill so suddenly from inhalational anthrax that there isn't time for a T-cell response. Natural killer cells can do a lot of the same things, and they can do them immediately. In test tube experiments, a collaborative team led by Professor Johnny Peterson profiled the natural killer cell response to anthrax, documenting how natural killer cells successfully detected and killed cells that had been infected by anthrax, destroying the bacteria inside the cells along with them. In further experiments, the group compared the anthrax infection responses of normal mice and mice that were given a treatment to remove natural killer cells from the body. All the mice died with equal rapidity when given a large dose of anthrax spores, but the non-treated mice had much lower levels of bacteria in their blood. This is a significant finding, said the professor. Growth of bacteria in the bloodstream is an important part of the disease process. The next step is to apply the existing natural killer cell technique to mice in an attempt to see if the more numerous and active natural killer cells can protect them from anthrax.
Is that the gangsta cells?
Mm-hmm.
That's a nice vest.
Thank you.
Next, we'll tell you how you can keep from getting an HIV infection after this commercial break.
Welcome back. Now we have tiny molecules protect fungi from the dangers of sex. Pathogenic fungi have been found to protect themselves against unwanted genetic mutations during sexual reproduction. According to researchers at Duke University Medical Center, a gene silencing pathway protects the fungal genome from mutations imposed by a partner during mating. This pathway was discovered in Cryptococcus neoformans, a fungus that commonly affects humans, causing over 1 million cases of lung and brain infection each year and more than 600,000 deaths. This discovery is how the genome is protected during sex might be leveraged as an Achilles heel in the battle against C. neoformans, which frequently causes life-threatening illnesses in people. Sexual reproduction in fungi produces airborne spores that are readily inhaled into the lungs and thought to be the source of human infection. Thus, agents that block fungal sex might stop the risk of infection at the source.
It's very interesting, Crystal.
Say fungal sex?
Fungal
Alright.
fungal sex.
Intravaginal practices may be associated with acquiring HIV infection. Some intravaginal practices used by women in sub-Saharan Africa, such as washing the vagina with soap, may increase the acquisition of HIV infection, and so should be avoided. Encouraging women to use less harmful intravaginal practices, for example, washing with water alone, should therefore be included in the female HIV prevention research strategies in sub-Saharan Africa. The authors pulled individual participant data from 13 prospective cohort studies in sub-Saharan Africa involving nearly 15,000 women, 791 of whom acquired HIV, and found that HIV infection within 2 years of enrollment in the studies was associated with self-reported intravaginal practices. After controlling age, marital status, and the number of recent sex partners, women who used cloth or paper to clean their vagina were nearly 1.5 times more likely to have acquired HIV infection as women who did not use this practice. Furthermore, the insertion of products to dry or tighten the and intravaginal cleaning with soap also increased women's chances of acquiring HIV. Intravaginal cleaning with soap was associated with the development of bacterial vaginosis and disrupted vaginal flora. Very interesting. 2 conditions associated with the increased risk of HIV acquisition.
That was interesting.
Yes.
Drip dry, ladies.
Very much. Well,
And
that's the microbiology news from Keenan TV.
so remember, America,
An acidic vagina is a healthy vagina.
This is WLMK, a Keenan television network. Have a good evening.